Hi, everybody. I'm so glad to have this chance to share my basic research using transgenic zebrafish. So I need to switch your mind from disease to a very simple model system. So first, I want to go through HPG axis, also called hypothalamic pituitary gonadal axis. So the group on we know gonadotropin-releasing hormone is very important in control of sex development and reproduction. The group of neurons in hypothalamus called gonadotropin-releasing hormone neurons. So those neurons produce gonadotropin-releasing hormone and then travel down to anterior pituitary, release luteinizing hormone and follicle-stimulating hormone, and then targeting on gonad and stimulating gametes produce, and also produce sex hormone in order to regulate sex behavior overall in control of reproduction. Because this group of neurons is so important, that integrate all the external signal, internal signals, and plus the regulation from the neuron, the other neuron peptides and neuron transmitters and the hormones in order to get reproduction control. So that's why we focus on this group of neurons to use a very simple model system to study this group of neurons development. Why we are using zebrafish? In recent years, decades, zebrafish become a very favorite model system for biologists and also for doctors. So the reason for that is because it's still called still vertebrates. Even very short, very small, but it's still vertebrates, close to human. And very inexpensive. So for one pair of female and a male, and one time they can produce like 200 to 500 eggs. So it's also very easy to maintain. The most important thing is the external embryonic development. So it's after the mother's womb. So you can exactly monitor the embryo development even when they are still embryo. So another very important feature is the embryos is transparent. So you can see organ generis, and also you can use the confocal microscope. You can follow the if you label the certain neurons or certain organs, you can see how they develop. It's very visually. And another thing is faster development. So in 24 hours, you can, from one single cell embryo, you can see a little fish 24 hours, and they have heartbeat and they can move a little bit. And definitely because they can amenable to genetic and cellular manipulation. So here, example, you can label whatever proteins and things you're interested in. In zebrafish, there's two forms of gonadotropin-releasing hormone. Also, we can call GnRH2 and GnRH3. In human, only GnRH1 in control of reproduction. So far, as we know, but zebrafish is a little different. GnRH2 is produced by a group of neurons in midbrain. It's so far the function of GH2 we believe is related to sex behavior. But GH3 in zebrafish is a main hormone in control of reproduction. Not only that, also there's a neuron modulatory function. The function of GH3 depends on where those neurons are located. The previous study from the other lab showing there's a few different groups labeled different colors here. These are terminal nerve GH neurons, and then here's the preoptic and hypothalamus, a group of neurons. Those neurons produce GH in control of reproduction. And there's a new group of neurons recently identified is in trigeminal neuron ganglia. So we don't know exactly what the function is, but we make that possible in the future we can study by making the transgenic GH3 zebrafish. In Embryonic is a variant of green fluorescent protein, like GFP. It's very super bright, and it's doable for the all kind of manipulation like immunochemistry in situ after many washes. Still will show very strong fluorescence. So this special model system allowed us to follow the neuron growth for a long time. And also we can use that for in vitro study. So using this model system, we did the morphological and the physiological study in vivo. So this kind of in vivo studies definitely is very limited for the other kind of animal model system, like rodents, of course, and that's impossible for the human. So the data I'm going to show you is 3 portions. One portion just to show you with this transgenic zebrafish how those neurons expression, and how these neurons form the network. And later on, I'm going to use electrophysiology showing you how we detect the neuron activities and the regulation. So this have a model, this expression of GIH-EMD. So this 40 hours embryo post-fertilization. You can see here there's nothing there in just the little fluorescent background. And here's the expression of this group of neurons. So you can see here's the terminal nerve close to olfactory bulb. Here's the eyes, retina, and then here's hypothalamus. And then this validation of our model system. This one is the expression of EMD, which present the gene H3 neurons. Here is using the antibody. We're showing the localization of these 2 different color labeling. So that means our model system is validated. So now, so this picture taken in live embryos. So at the 26 hours, we set the embryos lateral up, and you can see the whole network. Here's a trigeminal ganglia. So it's probably for you, as your first time heard GH neurons also could happen in the after the brain. And also here's the spinal cord. You can see the neurons, the multiple neurons here. This is a little later, 49 hours. So we focus on the head. So we set the embryo ventral up. You can see the multiple group of neurons. Here's the terminal nerve neurons again. Here's the retina. You can see many neurons expressed. And here is the hypothalamus. Because the bone is the little deeper, you couldn't see that clearly. Here's your yolk sac. So then we focus on different population of GNH neurons. We saw each group of neurons, the shape, the timeline is different. So here's the terminal nerve GNH neurons. Like I said, is associated to olfactory bulb. So here's a preoptic group of neurons and the hypothalamus. So these two group of neurons is believed in control reproduction in zebrafish. This group of neurons is play a modulatory effect. So and this one and the spinal cord, these two group of neurons is in the future we need to identify this new identified group in this model system. By fixing that, we can fix the embryos at different time point and then use a confocal to check the fluorescence expression. And then we found different group of neurons, they showing up at a different time point. The first showing up neurons is from the spinal cord and the trigeminal group here. And then later on is terminal nerve group and the hypothalamus. So these two group neurons, they show up about the same time. And later on, the retina and the preoptic area. So this gives you a rough idea. Using this model, we can identify the neurons at different time points. I'm from California, Los Angeles. So you know the Hollywood there. So I try to be a movie director, but taking this movie is not easy. So we have to follow the left embryo under the microscope. We take images every 30 minutes, and then we make a movie. So this group of we have a team to produce this very short movie. I hope you enjoy it. So now we focus on the trigeminal group neurons, 17 hours. This movie covers 17 hours to 24 hours time post-fertilization. How will I play? Okay. Oh, yeah. Okay, here we go. So you see the trigeminal neurons start appearing, one side and another side. And then they start making aftergrowth, and then connect to the spinal cord, and the more neurons coming out. And also the two bright spots, that's terminal nerve neurons. They start showing about 24 hours post-fertilization. So you can see dynamically how those neurons develop. So is that any question? So the next group, we are going to follow on terminal nerve neuron group. This one group of neurons, they show a little after. So how this group of neurons developed. This is 24 hours, and every 15 minutes we take an image. And you can see the neuron bodies appearing, and the neuron process also going out and withdraw, and here they are looking for where they are going. The neuron process is like a dancing. They try to find where they're supposed to go. So it's getting more complicated. Okay. So then we set the embryos like a ventral up. Here here's the whole embryo head, and the eye is supposed to be here. And this is terminal nerve group, this is hypothalamus. So we dynamically take images every, see from different, every 5 hours here showing. And we can count the neuron numbers. And then we can figure out how many neurons, like in certain time, have they grow. So here you can see the neuron numbers of the terminal nerve group. And after 40 hours, they stabilize, they got enough. And for the hypothalamus neurons there and the preoptic, they steadily increase the neuron numbers. We stop here, but the neuron number is still increasing. And here's another short movie showing like the whole group, the different, the terminal nerve group, hypothalamus and the preoptic neuron, how they grow together. So this cover about 12 hours. So start showing the two sets are terminal nerve neurons, and the bottom one is hypothalamus neurons. See the neurons at the red start appearing at the lower part, and they're getting more and more. And then now start showing the preoptic neurons. Some of them they show locally, and some neurons is from the hypothalamus. You can see the neuron process is still, like I showed you earlier, the terminal nerve neurons, the neuron process is going up and here, there, and I try to find where to go. Yeah. So from these movies, we can learn like we can learn how this group of neurons they develop. This is really seems very simple, but really so far there's no direct movies show how they developed. So it's really exciting when you see them how they do it. And second, I want to focus on the communication. So we found that there's multiple group of GNH neurons, and we found those neurons, they are not isolated. They make connections. Here you can see, you already saw this movie. So here the trigeminal neurons, and the other side, these two are terminal nerve group. So they just start to proliferate. And then here's the spinal cord neurons. At a certain point, they just connect to each other. Here's another example of terminal nerve neurons. And this one side and another side, and they make a connections each other. So here's the connection part. And then we saw there's more connections they are making later on after development. Here's the terminal nerve, and then here's the preoptic neurons, and then here hypothalamus neurons. So they make connections not only group to group, they make not only terminal to terminal, they also make the connections with the preoptic area. So I told you earlier, terminal nerve neurons, they play a neuromodulatory roles. But this group of neurons, they in control of reproduction. They make connections, so they talk to each other how they regulate in control of the whole reproduction process. And then later on, we use synaptic marker SV2, synaptic vesicle protein, label the vesicle to see the early synapse formation. So here's an example, like a trigeminal neurons at 25 hours. You can see the very beautiful colocalization on this group of neurons. That means the early synapse formation is already happened at 5 hours. So there's already synapse inputs on the GNH neuronal systems. And the next part, I'm going to focus on the electrophysiology. So we can use a fine needle to, in the left embryo, we can insert the needle into the neurons, and then use the setup to record the neurons' activities. Here's the showing like these electrodes, this terminal nerve neurons, and this is left embryo. You can still see the heart beating. And this is the close look of how these electrodes insert neurons, the multiple neurons here. And use fluorescence, you can identify, you record it from the red neuron. You can see the electrodes already suck some fluorescence protein here. And you can see beautiful here showing the traces, how the action potential look like. And when neuron firing, they talk to each other by this way. So you can see this is bursting neurons, this is tonic neurons. Tonic neuron like they keep beating, keep firing. So this is only 2 and 3 days left embryos. So here's the summary data showing 2 days and 3 days, the differences of the firing pattern. Means when they grow, when they develop, the neuron firing pattern also changing. So the tonic neuron represent the neurons more mature than the bursting neurons. They are learning how to firing. The last part, I just want to give you an example how we can use this to study hormones or other kind of factors, how they affect our neurons' activities. And also can do that morphologically. So here, kisspeptin, I think most people of you heard, is recently found neuron peptide is like a puberty gate, like a gate of puberty, where the neurons start firing and the puberty will start it. So here we just apply kisspeptin to see if that modulates gene entry neurons at a very early stage. So here's the bursting neurons, that means immature neuron. When you apply kisspeptin, there's no effect. You can see that this firing rate. But differently, if the neuron firing tonically, that's a more mature neuron, if you give kisspeptin, the neurons start firing more. That means at this stage, they start a response to this kind of neuron peptide stimulation. So summary, this transgenic model system is very unique because we have super bright EMD labeled this group of neurons, so allowed us to study neuron development at a very early stage. And you can study that in vivo. And we found out there's unexpected gene entry neuron groups, like I show you in retina and also spinal cord. You may think gene entry neurons only in hypothalamus. Actually, from this model system, we learn it's not only that. So in the future, more study probably will show in the different species. We'll find that probably is as much more complicated as you thought. So the multiple gene entry neurons, they likely forming a very complex network from head to tail. So they talk to each other to control the reproduction. It's not just the hypothalamus. So synaptic vesicles also, the expression suggests the early synaptic communication developed during the early embryogenesis, including in gene entry neurons. So finally, terminal gene entry neurons show spontaneous activities, even 2 or 3 days. And the firing pattern is related to neuron maturity and also affect on the response to stimulations. So finally, I would acknowledge our group. Dr. Wayne provide this opportunity for our team to do this kind of study. And we have a wonderful team. We support each other, especially during the midnight. We have to follow one of each other and then, work, switch our hours and try to follow the neuron growth. It's so fun, but it's kind of difficult in the night. Here's our campus of UCLA, and I welcome you. One day you can visit us and give your wonderful talk. Okay, thank you very much.
Do we have any questions? One right here.
Yes, hello. Thank you for the talk. I was a bit confused about the nature of the neuron firing, because usually in an adult, GnRH is actually released in the portal blood in bursts, right, by bursting activity.
Yeah.
But you were mentioning that this bursting activity represents immature neuronal firing, or the tonic one is mature?
That's a very interesting question. Actually, we are focused on different neurons. We are focused on terminal nerve neurons. Terminal nerve neurons is modulatory neurons. So the tonic firing is the mature firing pattern. The neurons in hypothalamus, they fire differently. They show different firing pattern. Yes. It's not only in this, they support each other, they also support the other tissues differentiation. The research from the other lab is already showing if they knock down GnRH neurons, also affected the eyes development.